I'm called Dr. Sisechityo Mtuluza, and I'm the Executive Director of Joint Clinical Research Center, also called JCRC. This is an international center of excellence that was established in 1991 by His Excellency, the President Yoweri Kaguta Museveni. And this was introduced as a scientific intervention in the HIV epidemic, which was at its peak in Uganda at the time. Since the inception of the JCRC, the center has led the pace in scientific innovations and research in the region. And we have been part of landmark trials that have informed policy and best practice in HIV prevention and treatment. The center, as part of this work, pioneered research on the use of antiviral drugs. These are drugs used to treat HIV in Uganda and sub-Saharan Africa in 1992. And because of the experience we had gained in the research and provision of these drugs, JCRC became one of the 2 case studies for the US President's Emergency Plan for AIDS Relief, which is called PEPFAR, in 2003. This program supports treatment for over 60% of HIV patients in low and middle income countries. And with the PEPFAR support, JCRC then led the rollout of HIV treatment in the country and also built capacity for other countries in the regions. And this rollout prevented senseless deaths that were occurring at the time. So in order to monitor the HIV treatment, we set up the first facility to measure response to HIV treatment by measuring the CD4 counts, or what we call soldiers in the blood, and also the level of the HIV virus in the blood as early as 1992. We established for the first time in 2004 the ability to determine if children were HIV infected as soon as they were born. Before this, we would wait until children were 18 months old before we tested them to determine if they were HIV infected. JCRC performed the first HIV vaccine trial on the continent to determine if we could prevent HIV infection, and also if we gave a vaccine to HIV infected patients on HIV treatment, we would be able to stop the drugs without having the virus multiplying again. These studies revolutionized how vaccine products were developed to include HIV types that were circulating in sub-Saharan Africa. And many trials have since been conducted in the region based on the work that we did from 1999.
Over the 30 years of JCRC's existence, it has carried out some of the groundbreaking research that has gone ahead to influence policy and guidelines up to the World Health Organization level. The center has continued to be at the forefront in research on newer drugs, assessing how well these drugs are working, how safe they are, as well as the possibility of being able to be given in low resource settings. Earlier studies like that study which happened in the adults, and another related study which was called the ARROW study that happened in the children that JCRC conducted, helped a lot to dispel the notion that ARVs could only be given in centers with sophisticated machines that are able to measure things like CD4, or what we call soldiers in someone's blood, and also the viral load, or the amount of virus in the blood. These 2 studies prove that actually ARVs could be given in the low resource settings like ours, where the machines are not readily available. A more recent study that JCRC has conducted, actually still ongoing, people are in the longer follow-up period, but this study has led to the understanding that treatment can be standardized for simpler dosing of an important drug called dolutegravir in small children with different weights. Initially, it required several different dosages for the different weights of children, which we all know that it naturally differs a lot per age. And so it has led to standardization of this drug and to be given in simpler form. But the same study has also showed that the drug, or the dolutegravir drug, can be safely given in smaller children weighing as low as 3 kilograms of weight. Earlier on, it could only be given to down up to 20 kilograms and above. In partnership with Janssen, and as well as the Viv Healthcare, which are pharmaceutical companies, and also with other partners, JCRC is leading and coordinating a study in sub-Saharan Africa on injectable ARVs in adults. This study is codenamed as CARES. It has a longer name, but we shorten it to CARES. And what this study is doing is that we are assessing how well this injection is working and how safe it is when these ARVs are given as an injection, which is given actually once every 2 months. Similarly, JCRC actually is also preparing for another study. This time it will be in young persons, that is adolescents aged from 12 years to 19 years of age. This other study is codenamed as LATTER. And this one will be starting actually in the first few months of the next year, that is 2022.
COVID-19 convalescent plasma has been used extensively in China, Europe, UK, and the US during the first and second waves of the epidemic. And what emerged was that for patients who presented early, if given convalescent plasma with high levels of antibodies, this prevented severe disease, hospitalization, and death. To collect convalescent plasma, we use a machine, an apheresis machine called a trimmer machine, and we use these kits that are connected to this machine. And for each patient, we use a single kit. As you can see, the kit is well sealed, and the kit inside here attaches to this area.
The same machine has been used for management of sickle cell disease.
In the clinic, clients are also assessed for eligibility for what is known as a red blood cell exchange procedure. A red blood cell exchange procedure is the removal of sickled cells from the patient and the simultaneous replacement of those sickled cells with normal red blood cells or packed red blood cells from the blood bank. At the JCRC, we acquired a machine known as an apheresis machine in 2017. This machine enables the simultaneous removal of the red blood cells and the replacement with packed red cells from the blood bank at the same time. This procedure is not curative for sickle cell disease, but helps alleviate persistent symptoms in patients with sickle cell disease who have trouble in controlling their symptoms while on treatment. It also helps prevent complications in patients with sickle cell disease. These complications, examples of these complications include stroke or even acute chest syndrome. In July of 2021, the Sickle Cell Clinic received a novel point of care test. This for testing patients or screening patients for sickle cell disease. This test is known as a Gazelle microchip electrophoresis platform. Electrophoresis is the procedure which we use, the modern procedure which we use for screening for sickle cell disease.
JCRC established a state-of-the-art laboratory that has attained the highest level of quality in the world. It is certified and is monitored by the International College of American Pathologists, CAP. The laboratory offers a testimony of over 60 tests, one of the biggest in the region. It supports both national and international research projects and has worked on several innovations to improve diagnostics of disease and its management. The laboratory was the first in the country to perform tests for monitoring to see if HIV drugs were working properly, including CD4 and HIV viral load. Following JCRC pioneering the use of HIV drugs, JCRC realized the need to closely monitor patients on HIV treatment to determine if the virus was developing resistance to the drugs. It established the first molecular laboratory in the region from 1999 to determine HIV drug resistance and support treatment. HIV drug testing is very crucial in guiding doctors on which drugs the patient is not responding to, which ones should be included in the patient's treatment regimen. JCRC laboratory is one of the 2 national laboratories that perform HIV drug resistance testing by the National HIV Treatment Program.
Okay, the laboratory has a laboratory information management system which manages sample data from the clinic system to when the lab result is released. The LIMS is integrated with the National System for HIV Drug Resistance, where results from the lab are transmitted in real time to the National Drug Resistance Committee. LIMS also has other functions that include tracking sample integrity, workload, turnaround time, among others. It also electronically assigns samples into freezers for long-term storage, and it can easily retrieve these samples whenever need arises.
The JCRC laboratory has a robust quality management system that is routinely monitored to ensure compliance to both local and international standards. As earlier mentioned, the laboratory is accredited by the College of American Pathologists, which accreditation status empowers the laboratory with multiple market advantages, which requires to maintain professional standards of services in order to stay on top. The laboratory undertakes 2 separate but complementary quality assurance activities that are integral parts to provision of reliable and accurate results. That is internal quality control, which is employed in each procedural step to ensure reliable, valid, and producible results. The laboratory also participates in CAP external quality assurance, which provides assessment of the lab result, accuracy, and staff competence in comparison with other testing laboratories or peers internationally.
The ionized water, also known as demineralized water, is the type of water in which all minerals, other chemicals, and harmful microorganisms, including toxins, that have been removed. It is very essential in all processes of laboratory testing and is required for quality results. We use it to import this very essential input to laboratory testing. We now produce our own deionized water for use at JCRC and also supply other laboratories in the country. This is our production unit. It consists of essentially ultra-clear reverse RO machine, overhead water reservoir tank, collection reservoir tank, and external filters. And this is our deionized water harvesting collection platform where we put water we have harvested, pending taking them to stores.
JCRC has 2 BSL-3 laboratories used to handle dangerous agents like TB, Ebola, COVID-19, or any evolving pathogens and emerging pandemics. The laboratory is established to handle in a safe way dangerous organisms.
While HIV viral testing remains an important part of HIV care, there are still a number of challenges associated with this test, ranging from prolonged turnaround time and the expensive nature associated with the test. So at Joint Clinical Research Center, we came up with a local innovation to address this challenge. We developed an assay named ESP, or exclusion-based sample preparation, which works as well as the conventional PCR platforms. Right now we are into field evaluation to prove that the assay works as well in the field, just as it was able to perform at the JCRC Center of Excellence.
The same platform has been adopted for innovations in COVID-19 testing.
As technology evolves, JCRC has acquired cutting-edge next-generation sequencing technology for early detection of HIV drug resistance mutations. This is important because it improves patient care, as it's able to pick out drug resistance mutations as soon as they emerge to avoid them widespreading. This technology is also very important in diagnosis of cancer. For instance, in hereditary cancer, we are able to identify genes associated with these cancers. For instance, some breast cancers and prostate cancers. This technology is able to detect mutations which could be associated with response to mutations. and also disease prognosis. This technology further on is important in identifying novel microorganisms and also identifying drug resistance in various other microorganisms.
So global blood safety is at the forefront of health focus. With these changes, it's time to take a proactive approach to protecting patients from potential dangers of donated blood. The Mirasol can also help you do it by reducing the pathogen load of a broad range of disease-causing viruses, bacteria, and parasites. The system also inactivates residual blood cells found in blood components, which may help to reduce transfusion reactions in patients.
During the outbreak of COVID-19 pandemic in the country, the supply of sanitizers became scarce and costs almost tripled. To ensure we got enough sanitizer for the institution to promote safety for staff and continuity of work, JCRC laboratory initiated the preparation of sanitizer. Currently, we are producing for home use in all our sites, which include Luboa, where we are right now, Mbarara, Fort Poto, Gulu, Chigezi, and Lango regions.
JCRC prides herself of a huge biorepository facility that collects, catalogs, and stores samples of biological materials such as urine, blood, tissues, and cells from humans, but can also store animals or plant materials if there is a need as we move towards One Health. Biorepositories are critical to enabling modern and future research, including genomics, proteomics, etc.
It has been a great journey with over 30 years of research and innovation that has informed both national and international treatment and prevention guidelines in HIV, but also other diseases common in the region. We've achieved many milestones and set trends in the way healthcare is provided. We'd like to say thank you to all our collaborators, nationally and internationally, who have supported us to undertake all this research and innovation work. We want to thank the government of Uganda that has provided an enabling environment and continue to support scientists to innovate.